Good morning, everyone. Today we are going to learn about one of important part in our metabolism as homeostasis, glycolysis, and gluconeogenesis. So what is glycolysis and gluconeogenesis, you may ask?
Glycolysis is the metabolic pathway that breaks down a glucose molecule into two pyruvate molecules, a process that generates a net of two ATP molecules and two NADH molecules.
In glycolysis, there is two phases, which is investment and payoff phase. Investment phase is named that way because 2 ATPs are used to invest, then payoff phase yields 4 ATPs, resulting a net of 2 ATPs. In the start of the first phase, which is the investment phase, glucose is phosphorylated by ATP and catalyzed by hexokinase to form glucose 6-phosphate. Then, the glucose 6-phosphate is isomerized to form fructose 6-phosphate, catalyzed by phosphoglucose isomerase. The fructose 6-phosphate continued to be phosphorylated by ATP catalyzed by phosphofructokinase, forming fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate will split into two 3-carbon molecules, dihydroxyacetophenone, DAP, and glyceraldehyde 3-phosphate, G3P.
Dihydroxyacetylphenone, DHAP, is then converted to glyceraldehyde 3-phosphate, which now results in 2-glyceraldehyde 3-phosphate. Meanwhile, in the payoff phase, glyceraldehyde 3-phosphate is phosphorylated by ATP, catalyzed by the G3P dehydrogenase to form 1,3-bisphosphoglycerate. Then, 1,3-bisphosphoglycerate transfers a phosphate group to ADP, catalyzed by phosphoglycerate kinase, forming 3-phosphoglycerate. 3-phosphoglycerate is then rearranged to 2-phosphoglycerate, catalyzed by phosphoglycerate mutase. 2-phosphoglycerate is converted to phosphoenol pyruvate by removing a water molecule. Lastly, phosphoenol pyruvate will be transferring a phosphate group to ADP, catalyzed by pyruvate kinase, forming pyruvate. Next, let's look at gluconeogenesis. Gluconeogenesis is the process of making new glucose in the body from non-carbohydrate sources, such as amino acids from proteins, lactate from muscles, and glycerol from fats. This process mainly occurs in the liver and sometimes in the kidney when blood sugar level is low, such as during prolonged fasting, starvation, or intense exercise. It is the reverse of glycolysis, but it uses special bypass steps because some glycolysis reactions are irreversible. The first bypass step is conversion of pyruvate to phosphoenol pyruvate. Here, pyruvate undergoes carboxylation to form oxaloacetate using the enzyme pyruvate carboxylase. Then, oxaloacetate undergoes decarboxylation and phosphorylation to form PEP, catalyzed by PEP carboxykinase. The second bypass step is formation of fructose 6-phosphate. Here, fructose 1,6-bisphosphate undergoes dephosphorylation to become fructose 6-phosphate using the enzyme fructose 1,6-bisphosphatase. Next, fructose 6-phosphate undergoes isomerization to form glucose 6-phosphate. Finally, in the third bypass step, which is the formation of glucose 6-phosphate undergoes dephosphorylation to form glucose using the enzyme glucose 6-phosphatase. The final product of gluconeogenesis is one molecule of glucose. To make this glucose, the body uses 4 ATP, 2 GTP, and 2 NADH molecules. To summarize, glycolysis breaks glucose down to produce energy, while gluconeogenesis builds glucose to keep energy available. Both processes are essential for survival, ensuring our body has energy whether we've just eaten or not. Thank you for watching.